The process of RNA synthesis inside a cell is also known as transcription, and the product of that transcription is oftentimes called a transcript. So if we start talking about RNA synthesis, what we're going to be doing during this process is we're going to be actually reading the DNA. So the DNA is going to serve as our template in this case. And because we're actually looking at the DNA and reading through it, this is definitely going to be taking place inside the nucleus. Now we're focusing on eukaryotic cells as we have been all semester, and at the end of chapter 17 we will compare and contrast a little bit with the prokaryotic cells. But for now we're going to be inside the nucleus looking at that DNA template, and this is a process that's going to be catalyzed by an enzyme known as RNA polymerase. That RNA polymerase is going to be really the worker molecule in all of this. It will be able to pry open or split the DNA strands from each other, and then it will also be the enzyme that's involved in hooking together or synthesizing the new piece of RNA that we're going to make from all of this. Now you do want to go back in chapter 16 and be able to compare and contrast RNA polymerase with DNA polymerase that we looked at in the last chapter. So if we just talk about some of the major points of similarities and differences between the two. First off, they are working on different processes. So RNA polymerase, this is going to definitely be on transcription, which is what we're getting ready to talk about. And DNA polymerase is going to work in the process of replication. Both of those processes do take place inside the nucleus, so they are functioning in the same location. If we talk about the template that they're using, we just said that RNA polymerase is going to be reading off of a DNA template, and it's the exact same thing for DNA polymerase. Keep in mind that DNA polymerase is looking at the old copy of DNA, synthesizing a new copy of DNA from that. Now, as far as the direction of synthesis, that will be exactly the same. So it's 5' to 3' in the case of DNA polymerase. It's going to be 5' to 3' for RNA polymerase as well. What they're making is different. So if we're talking about RNA polymerase, it is going to actually be making RNA. DNA polymerase is making DNA in all of this. Now to get the whole process started, there are going to be some differences there. So notice up here that it says that RNA polymerase is going to pry the DNA strands apart from each other. So this one, RNA polymerase, is able to open the strands. And keep in mind that opening the strands, by that we're meaning that it's able to break the hydrogen bonds. DNA polymerase required the help of an enzyme, and that was the helicase enzyme. So helicase was definitely needed in DNA replication to actually get the strands apart from each other. And then another thing that was necessary with DNA replication was that we had to have a primer. This one does not need a primer. This one can initiate. So if it can initiate, it's going to be able to add that very first nucleotide, and we don't have to have that primase enzyme involved. So it is going to be a simpler process than what we have with DNA replication. But keep in mind the molecule that we're making is not going to be really a long-lived molecule. It's going to be a temporary molecule that's going to tell the cell how to make a particular protein. In the case of DNA, that's a very stable molecule that is meant to last the lifetime of a cell. So if we go through now and we start talking about the steps of RNA synthesis, this is all inside the nucleus, and we're going to break it down into 3 different stages. And the very first one is called initiation. With initiation, one of the most important things that we have to do is we have to basically choose the genes. So by choosing the genes, we have to decide which genes inside the DNA do we really truly want to make a protein from at this particular moment in time. Keep in mind that human cells are estimated to have maybe 25,000 genes inside of them, and our cells are not utilizing all 25,000 of those genes at any moment in time. So a particular cell may be using 2,000 of them, or maybe 5,000 of them if they're really using a whole bunch. So all of the time, the cells have to really look through all of that DNA and they have to decide which genes are really the important ones right now. Which ones do we need to make into a copy of protein? So if we look at this, inside the DNA, we're going to have our double-stranded DNA molecule. Try to draw it as best I can here. It is anti-parallel, just as we talked about before. And all scattered along this piece of DNA will be little areas that we call genes. So this might be gene A, this might be gene B over here, and those genes are just sections of the DNA that are going to be able to tell the cell how to make a particular protein. So protein A or protein B. These are going to be scattered all throughout our 46 chromosomes. Notice that every little bit of the DNA would not really be what we would consider a gene. So we do have some little kind of spacer areas in between here. Sometimes it's called junk DNA, but it's definitely not junk DNA. It is very important DNA that's located there. So what the cell has to do initially is it has to be able to skim through all of this DNA, massive quantities, and really zero in on the genes that we want to copy into a piece of RNA. So to do that, there is a little section in the DNA which is called the promoter. The promoter would be a region just in front of the gene. So I'm going to just look at the B gene here. And this little section of the DNA, its purpose is to basically tell the cell when to use this gene. And when to use a gene, basically when to transcribe the gene or copy it into a piece of RNA. Or when to transcribe. There are specific features in the promoter that are going to tell the cell when they would want to use this particular gene. One of those features is what we oftentimes call a TATA box. Now this is called a TATA box because it is a series of T's and A's, usually TA, TA, AA, that is buried inside a bunch of other nucleotides that happen to be in that promoter region. If the cell happens to see the TATA box, then it knows that it is actually looking at a promoter region, and it should look closely because it wants to figure out if that's a gene that it might want to transcribe at that particular moment in time. Now, as far as the participants that are going to be looking at the promoters, these are called transcription factors. The transcription factors are small proteins, and they are basically scanning for promoters. So they scan through the DNA looking for promoters, and when they see a TATA box, they stop for a moment and they look more closely at the promoter to figure out when would this gene actually be useful to the cell. So besides the TATA box, there are some other sequences in the DNA that tell the cell other conditions when it would want to use that particular gene. There might be a sequence that tells it that that's a gene it wants to use when it's cold outside or when it's stressed out, when there's no food available, things of that nature. So these little bitty transcription factors are basically running around all over looking for promoters. When they find one and it looks good, they're going to bind to it. So we're going to have a whole collection of transcription factors that will bind to the promoters that look good. And as soon as they bind to the promoter, they're going to recruit or call over the RNA polymerase. Now, RNA pol is a very big enzyme complex, and that's really the reason why we need the transcription factors to be involved in all of this. RNA polymerase is not capable of scanning through mass quantities of DNA, so the transcription factors, they're tiny. They do scan through the mass quantities of DNA, and when they find something that looks good, they call the big enzyme complex over. And then the enzyme complex can get to work. At that point, many of the transcription factors go their own way. Some of them do still stick around with the RNA polymerase. But it is the RNA polymerase that's going to actually be making the RNA copy for us. So that's the process of initiation. We're getting the RNA polymerase to the promoter so that we can actually start making our RNA copy of the gene. Now the second step is what we call elongation. Well, in elongation, we are really making the RNA. So we do get the DNA strands opened up from each other. We're going to be breaking the hydrogen bonds in that case. And when we're transcribing a gene, we're not opening up a big replication fork. This time we're just opening up a small, tiny little section of the DNA at a time. It's actually a section that's going to be pretty much hidden completely underneath the RNA polymerase. And this means that it's going to be a lot easier to prop that little section open so that we can see the inside of the DNA and read the bases, determine the bases that we need to incorporate into the RNA molecule that we're making. So we're opening the strands. RNA polymerase is going to do that, and it begins to actually start adding the nucleotides, or put the first nucleotide in and then start adding additional ones on. When it does this, it is going to make RNA 5' to 3'. And when we make our RNA 5' to 3', we read the DNA in the opposite direction, so 3' to 5', because it's anti-parallel synthesis, just as we had with DNA replication. So if you look at this picture here, notice that the DNA strand, the blue that we're actually reading off of, this is the 3' end of it, and this would be the 5' end over here on this side. Notice that we're following the base pairing rules. C's and G's are across from each other. A's and T's are across from each other, with the exception that we would have the uracils inside the RNA instead of the thymines. They're also shown in blue in these, in red in these slides, the RNA nucleotides, because keep in mind that they also have a different sugar there. They've got the ribose sugar. So the elongation process is just going to continue along the way down the DNA strand. And all of this occurs until termination. Termination occurs when a termination signal is reached. The termination signal is a section in the DNA. So it's a certain stretch of DNA nucleotides. And this tells the RNA polymerase that it has gone far enough. So we only need to copy a small little region of the RNA into a piece, or of the DNA into a piece of RNA. Now I want to emphasize that the termination signal is not the same as a stop codon. The stop codon is something that stops translation, which will be the next process that we look at. The termination signal is what's going to stop the transcription process. When all of this takes place, the termination signal is reached, then everything just kind of falls apart. So that means that the RNA polymerase lets go of the DNA. It also lets go of the RNA copy that is just synthesized. And it can now be recruited to another promoter if the transcription factors have found one for it. Notice that we've got an RNA copy that is single-stranded. It has a 5' end and a 3' end, and that molecule is going to be much, much shorter than the DNA molecule is. DNA molecules are extremely long. We're talking about a couple 100 million nucleotides long in many cases, and our RNA copy is maybe 2,000, somewhere in that area, so much reduced in size. And we also want to emphasize here that this RNA copy is not what we would call an mRNA molecule yet. This is simply what we call a transcript. The transcript sometimes will get turned into an mRNA molecule, and we will talk about the steps that are involved in turning it into an mRNA molecule. But at this point, that really is the end of the transcription process. All of this is taking place inside the nucleus, so now we've got our RNA copy sitting there inside the nucleus. Now, if we assume that we're going to take that piece of RNA and we're going to turn it into an mRNA or a messenger RNA, there are some modifications that have to take place before we do call it an mRNA. First off, we're going to modify the ends of the RNA. Each end is going to get a specific thing added to it. On the 5' end, we're going to get a structure that we call a 5' cap. This cap is made from a modified guanine nucleotide. So it is different from the other guanines that would be inside the cell, so it is distinguishable as a cap. And then we're also going to add what we call a poly-A tail on the 3' end. Both of these additions are necessary if we're going to have a functional mRNA molecule. Now, the reasons that we would add these is, first off, these are going to help stabilize the RNA molecule. So if they're going to help stabilize it, then it's going to last longer, which means that we're going to have a better chance of actually being able to utilize it inside the cell or be able to read the information that's contained in it. The cap and the tail help export from the nucleus. The purpose of this RNA molecule is going to be to tell the ribosomes how to make a protein. Well, the ribosomes are not located inside the nucleus, so we do have to be able to get this molecule out to them. So the cap and the tail are going to help with that. And then also they're going to help the ribosome actually bind to the molecule. So all of these three things are extremely necessary if this RNA molecule is to ultimately serve its purpose inside the cell. We want to be able to make protein from it. If we're going to make protein from it, we need it to last long enough. We don't want it to be degraded. We want it to be able to get out of the nucleus. And we also want the ribosome to be able to grab hold of it so that it's capable of reading the information that's there. So the cap and the tail will be added. These will be added while we are still having this molecule sitting inside the nucleus. Now, additionally, we have some other modifications that take place. And a big modification is what we call RNA splicing. RNA splicing is basically cutting and pasting of this RNA molecule. Our RNA molecule has different sections inside of it. You can look here in this slide and see that we've got some sections that are shown as red, and we're calling them exons. And then we've got other sections that are shown in pink, and we're calling them introns. Well, these are just different sections of the RNA molecule, so they really are just nucleotides that are added together. But before we send this molecule out of the nucleus, we want to get rid of the introns. So we want to actually cut them out and remove them and then paste together what we have left. So in RNA splicing, we are removing the introns and we are joining the exons. So that when all is said and done, we're going to end up with this RNA molecule. Notice that is completely solid red. So it is made up entirely of exons. We've gotten rid of the introns completely. Again, this is going to take place in the nucleus as well. If we look at how this is going to take place, this is going to be performed by a big complex called a spliceosome. The spliceosome is important for recognizing where the exon ends and where the intron begins, and then also cutting the intron out and then joining those exons together. As far as what makes up the spliceosome, it is these particles called small nuclear ribonuclear proteins, or commonly called SNRPs. These are made up of some proteins, which is the purple component shown in this picture, and then they also have some RNA components, the snRNAs, which are called small nuclear RNAs. And the proteins and those snRNAs are going to be all complex together. Now, RNA, because it is single-stranded, is capable of taking on a lot of unique shapes. So you do see the red here is in this molecule is folded up in kind of a unique conformation. And the fact that RNA can take on those unique conformations is going to enable RNA to do a lot of different things. So this particular one is what we commonly call a ribozyme. This up here. Ribozymes are RNA molecules that function as enzymes. This happens to be the enzyme component of the spliceosome. It is made of RNA, so it's an exception to what we said before, where we said that enzymes are generally protein molecules. In this case, they are not. So we do have some RNA molecules that can function as enzymes. Well, what those SNRPs are going to do is they are going to actually recognize these little junctions right here. They're going to bind to them, and when they bind to them, they actually then come together, and as they come together, it's going to force the intron out in a looped structure, at which point we can really just cut here, and then we're going to really join these two together. So when all is said and done, we've got our RNA molecule that has no introns left in it at all. Now these introns that we did remove, those can basically be recycled. We can use the components to make other RNA molecules that we may need inside the cell. Now this splicing animation here is shown in your extra resources on Angel. So do take a look at that, because I think it will clear up what we mean as far as what splicing is, and you'll actually see the process in action. Now, there are a number of good reasons for splicing to take place. What splicing is going to enable the cell to do is it's going to enable the cell to take information in the DNA and rearrange it a little bit. So we can get what's called alternative splicing. Alternative splicing is an extremely large or big advantage of eukaryotic cells. This is not something that takes place in prokaryotic cells. So with alternative splicing, what's going to happen is the cell is going to be able to take the information in the DNA and maybe change the locations of the exons and the introns from one moment to the next. Well, if we change what we're cutting out and what we're keeping, then we're going to change the information that the ribosome is going to see later on. Keep in mind that the ribosome is going to make a protein from that particular template. So if the template is different, the protein that's made will be different. So in this case, we can now make many proteins, or many different proteins, from one gene sequence or one DNA sequence. And this means that although humans, for instance, have 25,000 genes, it does not mean that humans can only make 25,000 proteins. They can really make a limitless number of proteins, because they can simply keep rearranging the RNA molecule before it is sent out to the ribosome. So that is one very good reason to have introns present. Another reason to have them is that they basically expand the DNA sequence. So if they expand the DNA sequence, or in other words, they make it longer than it was before, the reason this is useful is that it's going to increase crossing over chances. Keep in mind from chapter 13 that crossing over results in new combinations. New combinations are extremely important for natural selection. If we have a very large region in the DNA, then we have a higher probability that when a crossover event takes place, it may take place within that region. So if we add little spacer regions inside the DNA, then we're going to increase our chances of actually getting some new combinations within that particular DNA sequence or within that particular gene. One last reason that we have introns is that these do sometimes contain regulatory sequences. And by containing regulatory sequences, that means that they are going to sometimes have things similar to what you would find in the promoter region of the DNA. So that means that if the transcription factors, for whatever reason, happen to miss the promoter, they can look carefully at the introns, and maybe then they will go back and decide to actually, yes, transcribe that particular gene into an RNA copy. So they kind of give the gene a second chance to be looked at.